to introduce my work today, and I appreciate Dr. Reza and Sue to invite me today. So please, first I'd like to share my screen. So can you see my screen now?
Yes, we will see.
Okay, so today I'd like to talk about the miniaturized water quality monitoring system. And as you know, the water quality is very critical issue in recently, and it is well known that even in the United States, multiple million people are suffered from contaminated water every day. So this water quality monitoring is quite a huge issue in even in 21st century. And maybe some of you remember the recent incident in Flint, Michigan, and in that city, the the tap water was contaminated by lead. So almost 100,000 people were suffered from the exposure to the lead. And that happened just back in 2015. And since that incident, EPA requested the lead testing for the drinking water to every state. So, and the results are quite shocking. So we could see that the spring water fountains inside the school and the many buildings are contaminated by lead. That means the consume when you consume those drinking water, you might be exposed to the lead. So that also enhance the this area to develop the water quality monitoring system. And currently, the best uh, way is using around 500,000 equipment, and then to detect the dissolved organic and inorganic materials from the water sample. And one biggest problem from that one is the equipment itself is very expensive, and even we can get very accurate and results from the equipment, and we can test multiple numbers and sometimes hundreds of molecules from the water, but we need some technician or some people who can operate the machine. So the one requirement related to this water quality monitoring system is the development of the some simple equipment that can detect the contamination very quickly and the location where it is required. So, for example, if you have a cheap device that can be attached to your faucet in your sink, and then the device can tell you every morning that the uh, level of contamination, that will be quite the best way to be safe from this contaminated water. So this miniaturized water quality monitoring system is one of my research area, and that is the one that I want to introduce today. And I want to start from the introduction of myself. So I'm Woo Jin Chang, and I'm an associate professor at the University of Wisconsin-Milwaukee. And I am affiliated. the- Okay, so I am affiliated mainly in mechanical engineering, but I'm also affiliated in biomedical and Great Lakes Water Institute, which is the unique institution in that is completely working on the freshwater area in the US and Medical College of Wisconsin. So you can see I have quite various affiliations from bio and medical to the engineering, and the reason is my background. So my background, uh, my PhD degree is from bioengineering, and then I was a postdoc in Department of Chemistry and Electrical Engineering, and then finally, currently, I'm in mechanical engineering. So the reason that I can do that is the trend of the current research area. So my lab is name is Biosensor and Biomems Lab, and the Biomems, this MEMS indicates the mechanical and electrical uh, systems. So because you can see the bio as well as some other research areas from this name itself. So it is quite multidisciplinary area. So this multidisciplinary means many uh, areas like chemistry, from chemistry and biology to many engineering fields like mechanical and electrical are integrated together to develop this kind of device. So I have an example shows the my work in here. So this picture shows the small device, and this one is actually just a 110 millimeter by 3 millimeter device. And then this yellow area is the electrode, and it is not quite clear, but this bluish area, it is the microfluidic channel. So it is the small channel that the water can uh, go move around. And in here, you can see the expanded view, and you can see the smallest feature is for this device, it is 20 micron. So uh, typically human hair is around 100 micron. So if your hair is thinner, then maybe 80 micron or something, and thicker, it is 120 or something. So you can see this size is just a quarter size of the typical human hair. And then uh, the purpose of device is handling the fluid inside this microchannel and then detect the electrical property of that liquid simultaneously and then multiple times. So as you can see, this device itself has the electrical engineering and the mechanical engineering field, as well as the target was the detection of the biomolecules. So this also connected to the biomolecules. So it is quite a multidisciplinary research area. And the reason that I want to do this will be explained from next slide. So I wanna talk today to briefly introduce the biosensor and biomems first, and then introduce my work. about the miniaturized water quality monitoring system. And for this, I have two approaches. And first one is electrochemical approach to detect the water quality. And second one is bioluminescence based, like a cell and bio reaction based system to detect live cell and toxicant. So I want to introduce this part more and very briefly talk about this bioluminescence based system. Okay, so yeah, I already introduced the example of this biomems device. So uh, you can think about the detection. So whenever you want to know the some amount of the chemicals or some features like glucose level in the blood, then there will be some sample, and typically it is liquid. And then for example, like this glucose level detection, you will just take some small amount of blood and then put it at the tip of the sensor. So this part is the sensor, and then this device read the signal from the sensor and decide how much glucose was included in your blood. So, this is a really great example, shows the goal of this biosensor and biomems device. So what we want to do is making this type of device and system so that anyone can do this measurement whenever required and wherever it is needed. And this pregnancy test kit is also a good example. And some difference will be this pregnancy test kit, you can read the result optically, just read the signal, and then you can decide. And this glucose device can get you the specific number of as a glucose level in the blood. So that is the slight difference, but these two are quite ideal example of the device that I want to make. And frequently, uh, the detection is not quite simple. So in that case, I need to use multiple samples and multiple steps to do the testing. So that type of detection is also available. And because of that, this type of device that can do multiple treatments required in this testing kit is integrated in one device. So from that, this concept of lab on a chip is generated. So basically, in these two examples, the detection was quite simple, but in typical applications, we need many different process to do the detection. So typically, we need to treat the sample first, and then make some reaction, and then some post treatments should be integrated, and then finally we can detect the level of certain molecules. So this slide just shows one example to do so from the sample of the biological cell, and then detect certain amount of DNA by PCR. But it is just a typical process to achieve this detection. So the question is, here is the my research interest. So question is how to get the amount of certain molecules. And in our case, the target is water. So, and the sample is water and target is the some contaminants inside the water. But it could be food or some body fluid. And I want to use and apply this biosensor and biomems technology so that this process required to treat the sample until obtain the quantity of the target molecule can be integrated in the single device so that the user does not need to do many things. Just the device can do everything. So user can just drop the sample, then the user can get the result. So that is my goal. And target molecules include some toxicants and some biomolecules. And well, today, in today's talk, I'll be more focused on these heavy metals only and pH and some briefly about the microorganism. And but I am also interested in these cells as well. So I have two directions in my lab. So first one is detection of these toxic molecules from water. And second one is the identification and separation of the cells. So, and because of that, last year I received a National Science Foundation rapid grant about the COVID-19. Uh, COVID-19. You know, the COVID-19 pandemic was caused from the virus, and I wanted to develop the method to separate and identify the COVID-19 virus from other nanoparticles and other less fatal viruses. And so I'm working on that project as well, but I would not mention that. I would not introduce that one, and I'll be more focused on these heavy metals and other toxicants. So the goal is identify the target molecule and then uh, quantitate how much that target molecule is included in the system. And also these molecules can be used to characterize and separate the molecules. So this is my research interest, and I wanna move on now about the water quality monitoring system. So yeah, I already mentioned about the importance of water quality monitoring. So I don't need to talk too much on this, but obviously water quality is very critical issue even in the United States in recent days. And I don't know, some of you may use the tap water, like municipal water, and some will use the water from the well. And I don't know how frequently you are checking the quality of your water. So I'm just expecting not many people do that because I had a project which is the National Science Foundation I-Core program, which supports me to meet people and interview them uh, about their experience related to the water quality monitoring and water quality. And then, interestingly, most people are not are interested in the water quality, but they don't do anything to confirm the quality of water that they are using. It's quite interesting. And even I met an engineer who is developing the water purification filter and the purifier system. And even the engineer does not check the quality of water at home. Just the engineer uses the filtration unit and then do not check the quality. So that is the, well, quite uh, current situation related to water. But if you are interested in the quality of water, you can just contact the water supplier in your area and then ask the testing results. They do the testing every day, like and every month, and it is open information. So you should be able to access that and see that. And one problem in current water quality monitoring system is the detection is done at the site where the tap water is supplied. So what it is, the quality is tested at the water treatment facility. And problem is the if you are living in old cities, then the probably the pipe system is contaminated and made from the material containing lead or any other heavy metals. So that the pipeline system is the major source of contamination. So even the water quality was good when the water left the facility, the water that you are drinking could be contaminated. So because of that, we need some system that can check the quality of water when close to the consumer, so that we can assure the quality of water. So that is the one major reason that the portable or just a small and cheap and robust water quality monitoring system is required related to the drinking water. So of course, there are some, uh, we have water everywhere. So as you can see, the natural water is also an important issue. So depending on the samples, we need to have different approach in water quality monitoring. But fact is, it will be always good to have cheaper device that can do this monitoring very quickly and on site. So, so instead of this like 500,000 device, if we can have something cheaper and do the detection quickly, then it will be a great plus for our safety issue. So that is the reason that I am developing this water quality monitoring. And so this machine is the mass spectroscopy to not only to detect the water quality, but can detect many inorganic materials from the water. And there are some different approaches to test the quality of water. And one interesting thing is, in our case, today's talk, I'll be more focused on the electrochemical water sensor that uh, are developed in my lab. And the bioluminescence based detection is connected to this biosystem. So basic idea is in the drinking water, this type of the device to detect the level of certain chemicals are enough. But in natural water, it is quite difficult to use this device because natural water has many different chemicals and even biomolecules and the living things inside. So pre-treatments are quite intense and important to detect. So it, one problem to use this type of detector is the time, because as I already introduced in previous slide, multiple steps to pre-treat the to do the pre-treatment, and then uh, some other requested procedure takes time. So this one always get you the kind of uh, result after a certain period of time. So it is not immediate. So some people developed some biological system to test the quality of water, in especially in natural water. So they put some small fish or small animal inside the water and then monitor their movement. So if the quality of water has some issue, so for example, toxicants are added to the water, then these animals will be dead, or at least the activity will drop. So by monitoring the activity of these animals, we don't know what the source is, but we can detect the we can monitor whether the water is has some issue or not. So that is quite interesting approach to continuously monitor the quality of water, and especially it is efficient to natural water monitoring. So the bioluminescence-based monitoring includes this technique, and I'll briefly introduce that at the later part of today's talk. And uh, here are some detection methods used in environmental monitoring. So, you can see the many different approaches like optical or electrical or chemical methods are used to detect certain molecules from the sample solution. And you can see that actually uh, almost any potential uh, techniques and technologies that can be used are applied to the detection. And every method has its own advantage and disadvantage. And I want to talk more about this electrochemical sensor today. So here is the diagram shows the detection of glucose from the sample. And well, there are many different ways to detect glucose, and some method just use the enzymes. And this one is the typically used method to detect the glucose from the blood. And actually, in this system, uh, we are not detecting glucose. So we are detecting hydroxy peroxide, not the glucose. So basically, we use the enzyme named glucose oxidase, and this can oxidize the glucose and then generate the hydrogen peroxide. And then at the surface of the electrode, this hydrogen peroxide is reduced. And then at the same time, we can measure this amount of electron to reduce this hydrogen peroxide. And then this amount of hydrogen peroxide will be proportional to the calculated electron. And then again, this is proportional to the glucose. So basically this electrochemical system to detect glucose, we use the enzyme reaction, which is bioreaction, to obtain the amount of electron, and that electron amount is correlated to the amount of glucose in the sample solution. So now then, by using this method, uh, get the we can calculate the amount of glucose dissolved in blood sample. So, well, you can you can note it can be noticed that this method can have advantages as well as disadvantages. So so if we can have a good system like this glucose oxidase, we can have very good sensitivity, which means we can detect very small amount of glucose, which is target molecule, because basically this electrochemical reaction amplifies the signal that we can obtain from the target molecule. So frequently, this electrochemical method can get us better sensitivity. So we can detect lower concentration of glucose with this method comparing with the method directly measure the glucose. So that is the advantage of this electrochemical method. And another advantage is the signal is electrical. So if you see this optical method, we need to use some camera or some photon counting diode or something to change this color into electrical signal. But in electrochemical reaction, it is always involved with the generation or consumption of the electron. So we can just simply measure the potential or some electrical properties from the related to the reaction. So that is another major advantage of this electrochemical approach. And of course, the one major disadvantage and or problem is we need to find the some better reaction system, because this electrochemical system use chemical or biological reactions to the amplify the signal we can obtain from target molecule. So we need the good reaction system to actually amplify the signal. So otherwise, it could be the sensitivity could be very low. So that is the one potential problem of this electrochemical system. And so this electrochemical system sensing is applied for many different target molecules of chemicals and biomolecules and so forth. And many different uh, reaction systems are used in this electrochemical motor sensor. And you can see, because in this electrochemical system and sensors, the, the the structure itself is quite simple. So here is the screen printed electrode typically used in many electrochemical sensors. And you can see this center area is the active electrode, and this circular area is counter electrode, and it is not displayed in this picture very clear, but there is a small reference electrode as well. So it is basically three electrode system that we can use. So we can have these three electrodes in the uh, detection. And that is the reason that you can see these three electrodes in this design as well. And because it is just the major issue in this electrochemical sensing is the development of the system, the reactions used to detect the target molecules, and that is typically treated the surface of this active area so that modify the surface of this electrode, then this sensor can be used to detect different molecules when the treatment system is different. So even the uh, format is the same, we can have different types of electrochemical sensor by changing the material to coat to modify the surface of this active electrode. And that is the reason that we can have these flexible sensors and then um, install these sensors to various materials like cloth and so forth. Okay, so uh, now I want to talk about the sensors that developed in my lab. And first one is pH sensor. So as introduced in here, I use this screen printed electrode and then use the different chemicals to modify the surface of this active electrode. And then depending on the treatment material, the sensor can detect different chemicals. So first one is pH. So to detect pH, I was using iridium oxide, and iridium oxide has these three different stages depending on the pH of the surrounding fluid. So basically, I just need to modify the electrode surface with iridium oxide, and depending on the pH, this iridium oxide status is varying, and then eventually the potential, electrical potential that this uh, iridium oxide have is varying. So by just measuring the electrical potential, I can decide the pH of the surrounding fluid. So it is quite simple method, and in typically this pH probe use the three electrodes and some solution inside to maintain as a reference, and also this one use some membrane so that the protons can pass through this electrode surface. But this simple printed electrode can replace this very complicated and expensive and even fragile pH meter. So it is quite a successful approach to make solid and robust pH meter that can replace this typical pH proving system. And in this example, the pH is correlated to the potential of the surface. So I just need to measure the potential of the electrical potential of the surface. So just a simple multimeter can be used to detect this potential. Just I need to have some power supplier and OP amp to amplify the signal, and that's it. So this setup is done in the lab, and that is the reason that I have these parts separately. But once it is done, then the completed device will be very small. And you can see this screen printed electrode and connected. Just the potential is taken from these two electrodes. And also you can see this white area. And this one is the just paper. So as you know, the paper has some very small structure inside. And the structure is consisted of the cellulose. And so it has micro scale as well as nanoscale pores inside. And then the pores are very random. So, and the cellulose are typically hydrophilic. So once the water is dropped, then it is absorbed to this paper structure because of capillary action. And so we wanted to use this paper, and you can see this circular area, which is the pattern written by some polymer, hydrophobic polymer, so that if you drop the water in this inside this circular area, just water will stay inside this area and does not go out from this area. So we wanted to use because the when we need to measure this pH, we need to dip this sensor inside the water. But by using this, we can just drop the just couple of drops of the sample, then we can do the measurement. And in many cases, the amount of sample is not super important. So let's say if you want to measure the pH of the drinking water from the faucet, then you have plenty of amount of the water. But sometimes it is quite important to have small amount of the sample. So for example, if you are producing the some let's say some drug candidates, some pharmaceuticals, and then typically the pharmaceuticals are super expensive. So easily like a 1 gram can go to a half million sometimes, something like that. So in that case, you don't want to waste samples just to measure the pH of the sample. So then in that case, you need to minimize the sample amount used to do the testing. So this paper-based system can help that kind of situation. And actually, the guy who approached me to test this device was working in the pharmaceutical area. So he wanted to minimize the waste of the pharmaceutical samples, and that was the major reason for him to test my sensor. So I compared the capability of the sensor using just a bulk solution, meaning the sensor was dipped inside the water and using the paper, and I could not see the much differences between this operation mode, I would say. And also this sensor was tested with different buffer systems and compared with the glass pH probe. And was again, I could not see much difference between the this screen printed electrode based pH sensor and the glass type uh, pH probe. So that was the first sensor that I wanted to introduce. And second one is heavy metal sensor. So see, now I cannot see the chatting box. So if you have any comments or anything, just unmute and talk. Okay, so the second part is heavy metal detection. So this is slightly different from pH. So regarding the pH, the structure is quite simple. Just modify the surface with iridium oxide, then and read the potential when the sensor is exposed to the sample. But heavy metals, it is a little bit different. And the approach that I was using is the method called anodic stripping voltammetry. And in this method, so we need to think about the sensitivity and selectivity of the sensor. These two are always very important features of the sensors. And sensitivity is the typically connected to the amount of target molecules that we can detect. So looks like a lower detection limit. So let's say whether my sensor can detect the EPA limit or not. That is related to the sensitivity. So obviously, we want to detect lower concentration of the target molecules as much as possible. So uh, typically, I want to detect like more than 10 times lower than EPA limit, and usually easily go to the 100 times lower. And selectivity is connected to the interference. So, so if you are thinking about the natural water has a lot of chemicals and molecules. So in our case, we just want to know the concentration of the heavy metals like lead or cadmium. But because many different molecules are also existing in the sample solution, uh, those chemicals can uh, interfere the detection in our system. So always question is how to detect lower concentration of the molecules even when the interfering molecules are existing. And the, the equipment cost like a 500,000. That one is expensive to purchase and operate because that can enhance the sensitivity and reduce the interference inside. So that is the reason that the cost of detection is rising. And so for heavy metals, it is same. So I want to detect lower concentration, but I want to avoid the interference from other molecules. So there are several approaches to achieve that goal. And in my case, I selected and I chose the anodic stripping voltammetry. And actually, this method basically initially deposit the dissolved heavy metals on the surface of the electrode by applying negative potential to the electrode. Because when the heavy metals are dissolved, typically they have cation charges, like positive charges. So if I apply the negative charge, that positively charged molecules will be attached electrically on the surface of the electrode, and then change the polarity of this applied potential to positive. And also at the same time, instead of just apply the certain potential, I can sweep the potential. So for as described in here, so you can see the potential applied to the active electrode can be swept from minus 1.2 to zero point 0.2. And then at certain potential, certain heavy metals are removed from the surface of the electrode. So if I can see the can quantify the height or area of this peak, then this one is correlated to the concentration of certain heavy metals. So this is the method used in this anodic stripping voltammetry. And the advantage of this method is I can have better sensitivity and better selectivity as explained in here. Because if I increase the time to attach dissolved heavy metals to the electrode surface, I can have more target molecules attached on the surface of the electrode, so I can have a better sensitivity. And the potential that certain heavy metals are removed from this electrode surface is different depending on the heavy metal. So I can detect multiple heavy metals single operation. So this graph shows the effect of deposition time, and you can see the up to 10 minutes of deposition to attach the heavy metals dissolved in the sample solution onto the surface of the electrode is rising up to 10 minutes, and it reaches to the maximum. So we can see like 10 minutes will be the optimum condition operation time to deposit the dissolved heavy metals on the surface of the electrode. And the next one, this slide shows the detection of multiple heavy metals in single operation. And in here, I mentioned that the potential of the target molecules removed from the surface of the electrode are varying. So because of that, okay, so because of that, you can see the multiple heavy metals can be detected in single operation. So this is the method that I'm using to determine the heavy metals. And also the this because I can adjust the deposition time, I can detect very low concentration of the heavy metals as well. So that is another advantage of this method, is that I already mentioned. And And okay, and I believe that you can see the annotations that I make on the slide. Please let me know if not. So, and
Yes.
okay, sounds good. Thank you. So, uh, the reason that I developed pH sensor as well is this one. The the heavy metals, the like this dissociation constant of the heavy metals are changing depending on the pH of the solution. So the pH measurement is kind of essential to determine the concentration of the heavy metals inside the sample. So typically it is fine, but if the pH is drifted significantly, then the measured concentration of uh, the heavy metals are incorrect. So for that, the both of pH and heavy metal measurement can be performed in single sensor as well, so that we can have better accuracy in heavy metal detection. Okay, so these are some uh, examples of the electrochemical detection that uh, I am developing in my lab. And currently, I'm working on the development of the commercial lead detector so that anyone can test the lead level in their drinking water. So that is the current status. And I want to very briefly introduce this bioluminescence detection because this one should be quite interesting approach to use microfluidics and bioreaction to detect toxicity and microorganisms. So in here, uh, I already mentioned that the toxicity of the water can be determined based on the activity of the biomolecules. And that is one method used to detect toxicity. And another approach is the detection of the microorganisms using bioluminescence. As you know, you may remember that we have frequently uh, we have problems from the contaminated food. So I remember that a couple of years back, tomato was contaminated. So E. coli was found. So it was recalled. And that happens quite significantly in even in recent days. And I remember that recently we had contamination issue in lettuce. So that happens. And the reason that the E. coli detection is important is not because E. coli itself is very fatal, but because E. coli is the indicator of the contamination. So that means if we can see the E. coli from the food, that means the food is contaminated. So that is the reason to detect E. coli from the system. And one problem is uh, we need to test whether that E. coli is live or not, and the amount of E. coli as well. So so it is always very difficult to check the, the live amount of live E. coli. So and it's not the perfect solution, but one approach is using luminescence. So there is a enzyme called luciferase, and this enzyme converts luciferin into another complex chemical, and at the same time, it generates the light. So, and thing is, when this luciferase is working, the ATP is consumed. And you know, the ATP is the energy source in almost all the living organisms. So if we have ATP only generated or extracted from the living organisms like E. coli, then this amount of light is proportional to the amount of ATP. So under certain condition that, let's say, if under the condition that we can expect no ATP is observed or included, then we can use this method. And if we have light, that means the sample is contaminated because ATP should not be included there, but it is detected. So this is one method to detect microorganisms. So I wanted to use this microorganism and the luciferase at the same time inside the microfluidic device to detect the toxicant and the microorganisms at the same time using the one device. So this luciferase is used to detect microorganisms, and this phosphorium is used to detect toxicant. Because this one is the microorganism generates the light typically like this, but when it is exposed to the toxic molecule, this light generation will drop. So same as measuring the activity of the live things, fish or a live animal to detect water quality, I can use this microorganism to detect toxic molecule. So yeah, obviously there will be several uh, hurdles to achieve this, um, to make this approach work. And I want to be a winner instead of these other guys. So because of that, I had to design kind of complicated uh, microfluidic device. And because both reaction use the light as a source, because the luciferase, the amount of light generated from luciferase will be connected to the amount of living organisms. And this light generation by phosphorium is also connected to the toxic molecule. So I wanted to add some time delay area so that I can first detect the generated light first to determine the amount of the living organisms, ATP, and then detect the amount of toxic molecule from this phosphorium strain. So I tested, and then the ATP detection limit was around 10 to the minus 7. And I can also detect the, the toxicant effect. So you can see the light generated from phosphorium is decreasing depending on when the concentration of the heavy metals are increasing. So I can successfully determine the concentration of heavy metals inside the sample as well. Just one uh, characteristic of this method is time. So because I need to use the bioreaction, the reaction time is kind of longer, could be longer than other methods, 5 minutes or 10 minutes. So, so that was about the bioluminescence-based detection of the simultaneously uh, microorganisms and the toxicants. Okay, so yeah, this my work has been supported from many different uh, organizations and under the support from many collaborators. Okay, thank you very much. Yeah, that's all for.
Yeah, thank you, Dr. Chang. Very good presentation today. Very interesting, um, designs I saw. So any questions from the audience to our guest speaker? You can type in the chat box also if you want. And also just a reminder, please complete the survey in the chat box to support this event. Yeah.
So I have a question. So thank you, Dr. Chang, for the very informative presentation. It's very great for us to start looking at different features of your research. So one question that I have, we will be working experimentally on ALD very soon with Dr. Shu. So one thing that I want to ask, what is the challenge that right now you can envision as it's mainly deposition techniques or finding the appropriate material? What is the challenge for the thin film deposition in your research?
Yes, so in the electrochemical sensors, the find out the best system, electrochemical system is the most important hurdle, because in electrochemical system we use the some biological as well as chemical reactions in detection, and there are a huge amount of different reactions from the chemical and the biological stuff are existing, and we cannot test like everything. So even I have the best uh, condition so far, that it is not guaranteed that that is the really the best condition that we can obtain. So there are still possibility that something better system is existing. That is the one hurdle. And yeah, obviously the surface treatment method is also a huge issue, because in my case, I had to use multiple numbers of different chemical treatments in series. So there are some possibility that every uh, fabrication, the consistency could be an issue in fabrication. So like a sensor to sensor variation is connected to the deposition condition. So in the lab it is fine, but in manufacturing, that issue has to be resolved to have a consistent lab sensors.
So I have a question about the when you mentioned detecting the heavy metals. So you showed different concentration. There are different curves. So is that so when you get the experimental data, you just basically go back and match the curve and see which, and then interpret the concentration. That is that the way?
Yes, right.
Okay. Okay.
But yeah, that part is
Mhm.
similar to any other
Mhm.
analytical methods.
Mhm. Yeah. So did you compare like your result with some like a conventional system? No. system, did you see like you know any difference in your?
Well, yeah, my system has quite good correlation. So for the heavy metals, the results are compared with the results from ICP mass,
Mhm.
and yeah, results are very quite close each other. So I could not see any statistically significant difference between my sensor and the ICP mass.
Okay, okay, very good. Yeah, because we developed something for just testing. We did some tests on some material that can potentially remove the heavy metal from water. So we just use ICP-MS, just did some testing. But I think your approach definitely is more interesting to kind of see how how it compares. And so yeah, that's fine. Yeah, very
Yes,
interesting to
right.
see that. Looks very simple, simple kind of
Yes,
concept, but I understand that the process, requires a lot of fine and tuning to accurately measure the results. But that's very yeah, interesting setup. Yeah. Yeah. Is there a question from the audience? I remember you mentioned something about your also interest in your research also in the wearable devices.
yes.
Can you like maybe share a little bit about that?
Well, yeah, because this electrochemical method can be applied for some biosample analysis. So I if the pH is also an important feature of the human body as well. So this pH sensor could be integrated inside the like a cloth or bracelet or something, so that the we can detect the pH level from sweat
Mm,
locally. So yeah, this type of approach is also applied. So we can find some like underwears or some other sensors that can be wearable by the human.
okay. Okay. Yeah.
Any question from the audience? So we see with Dr. Shu, we see, I mean, I'm not sure that if it's a long time that we talk, Dr. Chang, but as you know that Dr. Shu and I also, by the way, have the same research interests. We are working in the thermal fluid science. So some kinds of these components that you are describing, the CT, coefficient of thermal expansion, this mismatch is sometimes issue for our work. So I assume that you, first of all, the first question is that do you deal with these kinds of issue? Do you see this CT mismatch a problem in your research, in your work?
Did you say a CT mismatch?
CTE, coefficient of thermal expansion. So basically, if you are working with the temperature, at high temperature, the underlayer material that you deposit expand with different rate of change compared with another layer another substrate layer. So do you have this issue in your?
Uh, I think that one can be connected to the fabrication of the electrochemical sensor. And currently, I just use the room temperature to fabricate the sensor. So usually, I just need to drop the solution of the chemicals to modify the surface of the electrode, and then wait until it is drying in room temperature, and then drop second solution, and then wait drying. So it is multiple step, but I use just the room temperature. So far I haven't seen any issues from that, but I expect that if I need if I move on to the manufacturing stage, then I need to optimize the fabrication process. Then, I don't want to wait like 1 day in under the room temperature until the sample solution treatment chemical is drying. So in that case, I need to find the optimum temperature to do the drying. And then that temperature might be different depending on the solution, treatment solution. And in that case, that one will be an issue for me. So yeah, that one I should be I should consider in future.
Thank you.
Come.
Yeah, thank you. I guess maybe we can wrap it up here and thank our speaker. We will definitely be in touch about some possible collaborations and projects down the road. Definitely, I see a lot of common interest here. So thank you very much for the wonderful presentation today.
Hey, thank you very much, and yeah, thank you for invite. Mhm.
Yeah. I just want to remind the senior students in this class, please return back to our normal class link, and I'll meet you there in a couple of minutes. Okay, thank you.
Hey, thank you. Okay, bye.